So this month's scientific news. Robert F. Kennedy Jr. has been confirmed by the Senate as HHS secretary. So he comes into control of everything related to health in the United States. And as you know, he is a bit of an anti-vaxxer. And sadly, we just got also the first measles death case in 10 years in the United States in an unvaccinated child. Just announced 2 days ago that the CDC staff, maybe you can not talk, that the CDC staff will be prohibited from co-authoring papers with the staff of the World Health Organization. So that's not good. There was this article where there is hope that the policy changes might benefit the longevity movement, because Jim O'Neill, who also served in the Sense Foundation, was nominated to be the deputy secretary of the HHS, but he's still awaiting Senate confirmation at the time, as far as Wikipedia tells me. I didn't find any source that tells he's confirmed. The NIH has partially lifted the freezing on funding for biomedical research, because since Trump took office, there had not been any funding meetings, or at least not new ones that hadn't been scheduled before he became into office. And so they unblocked the first step of the funding, but not the second step. So, still no science can be funded at this time. Only the first step can happen, but then, before anything gets funded, you need to go to the second round, and the second round is not opened yet either. And also, very bad news is that the funding freeze is threatening the survival of the interventions testing program. So Richard Miller apparently in an email to someone else explained that the renewal is not happening. And yesterday PubMed was done for quite a long time, but I checked now today and it's back online. But yeah, that's very unusual that PubMed was done for many, many hours on end. And so I also published with a little bit of delay the February newsletter on longevity trends. And in the chat, I put a link to a Google form where you can subscribe to the email newsletter. And you can also subscribe to the journal club that me and Walter here present are hosting. And Stephen, but I think Stephen isn't here, but he's also part of the no, I'm being called, sorry. Yeah, sorry, where was I? Yeah, the newsletter and the journal club, so you can subscribe there.
We can only subscribe to one or another. It doesn't let me subscribe to both because I just tried it.
Oh, it doesn't allow you to click both options at the same time.
So I have to sign up with a different name and different email address.
Oh, sorry, I will fix that after the meeting. Okay, thank you for the feedback. I wasn't certain about that. Okay. Yeah, and there is the upcoming Dublin conference that I will attend. So if you are there, I see you there. So here, this paper, I think it was already in a discussion on the HEALS mail list. I saw something passing by between Didier and Aubrey about the impact of environment versus genetics. And I think this was probably the paper it was referring to, but I haven't just verified that. Yeah, anyway, this paper used UK Biobank data to look at environment versus genetics for aging.
Yeah, just saying, yes, it is about this, that there was a discussion between Aubrey, somebody else, I think Walter and me.
Okay. So yeah, so they derived polygenic, so multiple gene risk scores for 22 major diseases. And they found that they explained less than 2 percentage points of additional mortality over age and sex. whereas exposome, so exposome is all the environment you are exposed to, so everything from lifestyle to air pollution to even socioeconomic status and so on. Although I don't know how this paper exactly defined exposome, there might be slightly differences between how people define it. That explains 17 percentage points, so much bigger. However, of course, it depends from illness to illness, and so certain illnesses have a higher proportion being explained by genes, while others are more explained by the environment. Then there is here, and this was already in my newsletter, the Whitehall 2 study. So this is from Tony Weiss Correa, who has published multiple papers on single organ proteomic clocks. And here he applied those to government employees from the UK who were middle-aged at the time of recruitment, and then they had like 20 years follow-up done. And here they found that large organ age gaps were associated with an increased risk of 30 diseases. I mean, of course, this doesn't mean there are only 30 diseases. It's only 30 diseases for which they could demonstrate this, because of course they are limited by, for example, the number of people involved. You know, for some rare diseases, you might need bigger sample size and things like that. And so 6 of these 30 diseases were exclusively associated with accelerated aging of their respective organ. So that means, for example, liver failure was one of those, means that only aging of the liver was associated with an increased risk of liver failure, while, for example, aging of the heart wasn't. And then they had 24 diseases were associated with more than one organ age gap. So that means if you're aged in 2 different organs, including the organ in which the disease is happening, of course, that this was associated with the disease. And then there were another, 6 diseases where the risk of multiple other organs contributed to the disease, but not the organ in which the disease actually is happening. So I think here they had, for example, brain aging was associated with immune aging, but actually not with aging of the brain itself. So here there are 2 papers side by side published on FGF21 on thymic aging. So the thymus gland is very important for the immune system. And both of these papers show that if you elevate FGF21 artificially, you are protected from thymic aging. And so this extends the half span of the animal. And here in this paper, they also show the role of mTOR signaling in downstream of FGF21 in regulating the thymus function. Here they did infusion of young donor plasma in a randomized clinical trial. The results don't show much, primary outcomes, but they are mostly looking at differences in, signaling events. So here they have the Do Health trial, where they looked at vitamin D, omega-3, and exercise on DNA methylation clocks. And they find that omega-3 alone was able to slow down the pheno-age, the GRIM age. So pheno-age is phenotypic aging. GRIM age is the clock, the methylation clock that is trained on mortality data, and the Dunedin pace clock. And all of these were slowed down by omega-3 alone. And if you added to that vitamin D and exercise, you got additive benefits on pheno-age aging. Here also diet. I'm not going to go over that. Here they demonstrate that GDF15 is a blood marker of mitochondrial health. And here they show that when oocytes in a model.
Switch on the Wi-Fi.
Wait, what are you saying,
I
Anton?
see your. It's by accident.
Oh, okay, okay. So here, what they do is that as a mother is in her 20s or 30s, and the oocytes have been there from the very early life. So these oocytes are 20 or 30 years old when the woman gets a child. But so the mitochondria in those are also old, and apparently just before the oocyte becomes a zygote and so becomes available to be fertilized, there is actually a period in which there is increase in mitophagy, which actually removes the damaged mitochondria and provides a pool of healthier mitochondria. And that's the way in which the mitochondrial health is reset in these oocytes. Yeah, let's skip that. That's highly. Okay, so here in this paper, they look at a plasma proteome from 71 people between 20 and 100 years old, and they identify a bunch of hub proteins that are associated with aging. So hub proteins are proteins that show a lot of correlations with other proteins. Here they looked at an individual. I haven't checked who it is, but it's very easy based on this information to find who this person exactly is, who lived to 117 years of age. And so they, what did they all study here? Yes. So they had genomic data, transcriptomic data, metabolomic, proteomic, microbiolomic, and epigenomic data from that person. And so in this paper, you can find all that information. So here they show that lipids can actually contribute to Alzheimer's disease by causing by triggering the formation of amyloid beta aggregates. And here, very similarly, nanoplastics may also do that by triggering amyloid aggregation. Here, they took mice that were germ-free, and they reintroduced Escherichia coli in those mice, and they found that this actually caused amyloid pathology and cognitive decline in of course, this Alzheimer's disease model. Yeah, that's a database of Alzheimer's-related genes, so not much to say about it. It's just a resource. Here they find that a particular type of neurons, Purkinje cells, that they are showing a different vulnerability to aging depending on the location. So they are not just all are randomly susceptible. They seem to be like certain specific regions that come in stripes are more susceptible to dying, these neurons, than other areas. Iron was already well known to be related to Parkinson's disease. And here they also looked at two other brain diseases, multiple systems atrophy and progressive supranuclear palsy. And in both of those, they also find that iron is increased in the respective brain regions where these diseases occur. And here they look at the loss of these dopaminergic neurons in Parkinson's disease. And what they show is that the alpha-synuclein aggregation actually impairs the pentose phosphate pathway, which is a pathway that is important in the production of NADPH, which is a reducing equivalent, which is needed as part of the redox system in the cell. And so that leads to a loss of this redox balance, and that results in the oxidation of dopamine, which is well known to occur in Parkinson's disease. So this links the accumulation of alpha-synuclein aggregates to dopaminergic oxidation. So different important parts of Parkinson's disease get linked together in this study. Here they develop a mice model for osteoarthritis. Here they look at 3 antioxidants, and one of them, luteolin, was successful in preventing hair gray growing. So the graying of the hair, I should say, in a mice model. Both if this was applied externally or also if it was internally, so supplemented to the diet of these mice. Here, this paper looked at 41 skin phenotypes of aging. So aging is more than just wrinkles or pigment spots. For example, they found that telangiectasia, which is this red spider veins, these tiny spider veins, poor lip fullness, lighter skin color, cirrhosis, freckles, ptosis, so that's droopy eyelids here, eye bags, low eyebrow position, and so on were also part of skin aging. And they accounted for 22.5% of the variance in skin aging phenotypes in the population, while the wrinkles, pigmentation, and photoaging was 24%. So it's almost the same as the things that people think more about as skin aging. So there is a lot more to skin aging than just wrinkles and photoaging and pigment spots. And here they identify a whole bunch of genes that were related to skin aging. Here they show that topical ABT 263, which is a senolytic drug, actually improves skin aging, skin wound healing, I mean, in old mice. Yeah, this is a paper. It doesn't seem to have any data in it. It's more how it will be set up, the dog aging trial with rapamycin. So it's a background on that. Here they show that ginkgolide B, which is a compound derived from Ginkgo biloba, the tree, improves lifespan in mice when given at 20 months of age, so quite old mice already. In female mice, so I don't think they did male mice because it's not mentioned in the abstract. And they also showed that this improved muscle health in these mice. Here they show that in old mice, there is a deficiency in the catabolism, so the breakdown of branched chain amino acids. And this then leads to the accumulation of these branched chain amino acids, which activates mTOR, and that then leads to promoting sarcopenia. And you might know there is a clinical trial in humans ongoing or going to go on with rapamycin to prevent sarcopenia. Here they looked at a wild mice, Mus spratus, compared to the common laboratory mouse, Mus musculus. And what they find is that in this Mus musculus, the common lab mice, they which are adapted to human environments, they had weak acidification of the lysosomes. So that's very important for the lysosome to do its function to degrade stuff. It needs to be acidic, and that's not working as well in these Mus musculus. And they have high levels of beta-galactosidase, which is a classical marker of cellular senescence. And it's quite the opposite in this wild mice, which had much more acidification of the lysosomes and had less of beta-galactosidase levels. So does this mean less cellular senescence? Well, that's not entirely clear, at least not from the abstract. This paper was quite interesting because what they did here is they actually looked at regulatory toxicology data, and they looked at pesticides, and they took something like 36 or whatever pesticides that they studied, for which there was lifespan data submitted, and quite a few of these pesticides were actually extending the lifespan of mice. Much more than you will expect by pure accident. If you compare it, I think it did better than typical longevity drug screens. Yeah, skip that. So here in Drosophila, in fruit flies, there is the toxic Y chromosome hypothesis. So we all know that typically males tend to die earlier than females. And one possible explanation is that the Y chromosome has a lot of these transposable elements, which are also sometimes called jumping genes, right? That can move across the genome and promote genome instability. And yes, fruit flies do, or male fruit flies do have more of these transposable elements. And so this may indeed contribute to the shorter lifespan of males. Nicotinamide riboside is improving testicular aging, and it's also protecting against abdominal aortic aneurysms. So an aneurysm is when the blood vessels swells up and can actually burst open. So the way this works is that the production of collagen requires quite high amounts of proline, and NAD is important in the synthesis of proline. And so it may be that if you have a lack of NAD, you might have a deficiency in the synthesis of proline, and thereby a deficiency in collagen production, which may weaken the vessel wall. This paper substituted ordinary table salt with low salt or low sodium salt, so the potassium chloride version. And very interestingly, here recurrent stroke was decreased by 14%. Hemorrhagic stroke was 30% down. There was a 12% decrease in death rates of people who used that low salt. And stroke-related deaths were also decreased by 21%. So quite significant benefits from a very simple intervention, which is just buying low sodium salt versus ordinary salt. Yeah, this is just talking about the fact that multiple clinical trials do not use the same primary outcomes for assessing atherosclerotic cardiovascular diseases. And, you know, yeah, that better standardization may be necessary. Here we look at the oldest tortoise, Jonathan, and they found that various pathways associated with aging, such as DNA repair and telomere regulation, were different in Jonathan compared to other tortoises that don't live so long. Yeah, I think let's skip a little bit. Yeah, that's the C. elegans. We skip that. Reviews, again, you can look at that yourself. Here they develop a chemical way to reprogram cells to human pluripotent stem cells. And finally, here they show that COVID vaccination in children is associated with a 57% reduced likelihood of one post-COVID syndrome symptom, or with a 73% reduced risk of 2 or more of these post-COVID symptoms. So COVID vaccination protects very much against the long-term side effects of COVID. And that's the scientific news. Questions?
First, thank you, Sven. It was, like always, interesting. I have already a few questions. First, you said, like you know, there is a test for rapamycin and I forgot Alzheimer or Parkinson. I must say, I didn't know.
No, sarcopenia.
Sarcopenia, sorry. Yeah, I didn't know about this. Maybe you can tell a bit more. Then
I think it's in New Zealand, or so. I think I even shared it in the scientific news a few months ago,
okay.
and they were at that point in time recruiting people to participate. And I haven't checked if that trial is now already ongoing or still recruiting patients.
Okay. Thank you for the for the information, even if it is a bad news for the test on mice in the US there, it would be one more negative aspect. This is really quite kind of very depressing. I had one. Just I want to say, repurposing regulatory toxicology safety data. That's very interesting. That's something I'm thinking about that since many years. So I will read that attentively. And
it's a preprint, so it's available for free. You can.
Okay, good. And then something I did, I'm not sure I understood. So they made a randomized clinical trial for salt. So they were really asking for some people to take one sort of salt and for others to take another sort, or?
Yes, I think it was indeed a randomized trial. Yeah, a randomized clinical trial. So yes. I don't know if it was blinded or not. You know, you can kind of, I don't know with the mixture of salt with potassium chloride, but if you take pure potassium chloride, you can actually taste it slightly different. It's a bit more bitter than ordinary sodium chloride. So people might have guessed it's thing. So I don't know how blinded it was, but yeah, if you put it in your food, you will not know the difference. But if you taste it by itself, you could probably differentiate.
But I mean, it's a big.
It's a very big difference. But this is already this is already known for quite a while that you know, if you replace sodium chloride with potassium chloride, because potassium chloride actually lowers your blood pressure, right? Sodium increases your blood pressure, potassium lowers it, and so this lowers blood pressure. And it's very well known that this has beneficial effects.
Yeah, no, but my comment was more about the size of the.
Yeah, it is a very impressive effect. Yes,
600 villages. I mean,
and it's something you can you do without actually, you know, it's not much more expensive. It's it doesn't take any effort. You know, you just need to simply use a different type of salt.
Well, wait a minute. I have to stop that for a second. So this wouldn't be the same as just reducing your salt intake by 25%, would it? This is actually a combination of two things going on. One, you're taking less salt, and two, you're supplementing with potassium.
Correct.
Is that correct?
Yes, that's
this
correct.
is the title is misleading, I think. It's not really salt substitution. It's that's a misnomer. It's salt reduction plus potassium supplementation is what's going on.
What I mean is substituting regular table salt with that low low sodium salt.
Right.
So it's that's what I mean with substituting, right? Because you have like a bottle of salt and it's another bottle of salt, a mixture of sodium chloride and potassium chloride. So that's what I mean.
Okay. Anyway, this is interesting, and I will. It's also freely accessible, you think?
I don't know this on top of my head if it's freely accessible.
Otherwise,
and of course, that low salt is already available in the stores for quite a long time, so you can just go in the supermarket and buy this.
Yeah.